In my undergrad, I was unaware, as many people are, that fruit flies have brains. When I do outreach talking to elementary school or high school students, it's normally the first thing that I say, because no one thinks that an insect would have a brain. A lot of people didn't think about it, and even a lot of my family were confused because they knew I always wanted to study the brain, so when I joined the fly lab, they were very confused.
You might look at a fruit fly like in your kitchen or in the farmer's market and think that a fruit fly doesn't have anything to do with us, but actually a lot of the genes are conserved between fruit flies and humans.
Drosophila have a lot in common with humans. They have brains and guts and hearts and all that, but more importantly, for all the disease genes that we know about in humans, nearly 80% of them are identical in flies, and I think that that's what makes it a really good model, is that we can still make the same mutations in flies as we find in humans.
The things that we're able to see and the number of ways that we can see it with a fruit fly are not to be overlooked. I came into the lab to really learn how to apply what I already knew about flies to directly impact human health and human behavior too.
We can learn so much, especially with how closely related the genes are, and just figuring out how our brain develops.
So I'm really interested in studying neurodegeneration, which is basically like diseases that will arise in usually older individuals. And it's just a slow degeneration of the brain over time. And so this includes things like Alzheimer's, dementia, Parkinson's. And so I came to study flies because they're actually a wonderful model organism for studying these types of diseases. What we have in a fly is very similar to like how humans work, both genetically and molecularly. In addition, a fly has a very reduced lifespan, which is really important when you want to study these later in life diseases. So a fly will live about 60 days compared to like 60 years in humans. It's a very, very short lifespan, so I can do a lot of different tests and experiments on these flies, all within about 2 to 4 months.
Microcephaly is a rare neurodevelopmental disorder, so it's not super common, and it is defined by if a child is born with a head circumference, so they measure around your head, if it's smaller than 2 standard deviations or more, so their heads and their brains aren't developing properly or enough. They tend to have other impairments like inability to move properly, experience sensory input like vision and hearing, and they normally have intellectual disability and other sorts of accompanying symptoms. So I'll dissect out larval brains, so before they're adults, and I stain it with various proteins so I can see that if there are any changes in the brain. Because otherwise it's kind of a transparent tissue, so we have to stain it with different things to see it better. So this is just the brain lobe itself on the imaging, so you can see it kind of starts up here and goes all the way around. And this is just at one section within the brain, so again it's a 3D structure, and so we kind of slice it up like a sandwich when we're imaging to see all the various layers, because there's going to be cells throughout the whole brain. And so the staining that you're seeing here is the pink are the neural stem cells. So these are all of the cells these are the cells that are going to make all the other cells in the brain, the neurons, the glia, everything. So we have them stained because they kind of line the periphery of the brain, and this allows us to get a full reading of the volume of it, so we can look at size changes. For microcephaly, a lot of the genes that we already know that can lead to microcephaly if mutated are generally those that involve cell division. So if the stem cells can't divide properly, they can't make cells. The white staining is a marker for mitosis, which is cell division. So this tells us that these stem cells are actively dividing and therefore making other cells. So in this image particularly, I was looking at if there's any increases or decreases in cell division of these stem cells with the various genetic mutations that I was looking at. And this will tell me whether or not that my gene is necessary in cell division and give us an idea as to how mutations in this gene could lead to microcephaly.
looks like a good one. It looks like both a brain and a ventral nerve cord. A lot of my work is very exploratory, so I've already seen a lot of really cool results. I found that my protein can, when it's mutated, when this gene is mutated, it actually does cause a large reduction in like the fly's ability to climb and to fly. And yeah, it was all really exciting to see because I've actually figured out what specific cells this gene is functioning in. And this is incredibly key to anything therapeutic-wise. In order to help patients that have these mutations and have these diseases, we have to know what's going wrong in the body. So we need to know how it functions normally and how these genes can be disrupted in order to create viable therapeutics in order to help patients. Okay, so this is my setup for the flying assays. The first step is I have to get these flies off of food and into a clear vial in order to put them to sleep. For this assay, we have to use ice instead of CO2 because CO2 can actually affect a fly's behavior. Here we go. As you can see, I have now placed them onto a white surface so I can better see them. What you're seeing right now is me trying to get one to stick onto a needle point. So I'm not puncturing the flies with the needle or anything like that. I put a small dab of glue on the tip and then I will place it onto the thorax of the fly. And once it's on that upper thorax, then I can place it onto a little wall so that the fly is suspended. And then from there, we wait for them to wake up and then we can measure how long they fly for. And doing that kind of shows us if there's a significant decrease in their flight ability, which kind of equates to motor function.
So first, we rip the larvae in half, and then take my forceps and poke the head through the body. There's a lot of fat that the larvae have. This is part of the digestive system that I'm pulling away. And the brain is right here.
And so this is what a third instar brain looks like. So it's at that stage right before pupation. And it has two brain lobes, which you can see up here at the top, a left and a right one. And that's what is going to be more synonymous to our brains. And then it has a ventral nerve cord, which is going to be more synonymous to our spinal cord. And you can actually see these cool projections coming off, which are the axons from all of these neurons that are going off into the muscles of the larvae to help it move.
Larvae come in different sizes and shapes and therefore ages. This, for example, is a third instar larvae. This is the stage we actually take them at. That guy right there is a first instar larvae, a younger stage. This is the head end of the larvae, so where the brain is. And typically we can see they have these spiracles. You can see them on the pupae actually, and they'll stick out, and that's how we know that they're ready. So what we do is we pull the brain from this end of the fly as opposed to the other end. And sometimes I feel bad. Sometimes I'm like, I'm sorry, thank you for your service. And then I pull their brains out.
This right here is an adult Drosophila brain. We can dissect out the brains of these flies using fine-tip forceps. And so what you're seeing is actually a very, very zoomed in image of this brain in a very tiny well, like that big. And it's actually floating in PBS, which is just like a buffer solution we use to dissect in. So, as I mentioned, this is the adult brain. What's really interesting about fly brains is that over half of their brain volume is actually just the optic lobes. So you can kind of see that this here is the central brain region, like that. But then you'll notice like these kind of like dark lines, those actually separate out the optic lobes. So this is just one optic lobe and the other one's right over here. So I'm sure that everyone has seen a fruit fly flying around in their kitchen at some point, and you'll notice that they have these large red eyes. That's actually, there's a piece of an eye right here. I wasn't able to completely get it off in the dissection, but you can already see like that's the red color that you would see on the outside of their eye, but it would be over on this side of the brain normally. So those were the optic lobes, and then this right here, as I mentioned, is the central brain region. This is what I usually study when looking at neurodegeneration, since it isn't involved in the eye, it's involved in normal neuronal processes.
So right now I'm just setting up. I usually fill every single well because I'll dissect in one, I'll keep the brains in another.
All right, so this here is a video of me dissecting out an adult Drosophila brain. So as I mentioned before, we do it in these small little wells, and we use fine-tipped forceps. So the first step that I do is I actually, after these flies are anesthetized, so they are put to sleep, I will take the brain and separate it from the body. The next part is honestly the hardest, and that's what's happening right here. Flies have a outside cuticle, so they have more of like a harder skin than us humans have. And so you have to be really, really careful to take out the brain without squishing it or like puncturing it, but you still have to get through that outside cuticle. So the way we do this, as you see, I kind of already did it. You put them so the head is on its back, and then you can go up from the bottom, and usually that's a lot of a softer area. So I can, I start by putting the forceps like through that small skin area, and then I like separate like that, and that will usually separate the cuticle while leaving the brain intact. From here, you can see I'm doing it now. I slowly start to take off the cuticle until the brain is exposed, and eventually it'll separate from the rest of the head. And you can see it already poking out there. It is white. We use these really powerful lights in order to better visualize the brain when we're dissecting. It kind of causes it to glow a little bit. So the glow you're seeing isn't natural, it's just caused by the lights illuminating it.
People are very similar in their genetic makeup, but we differ in things like height and eye color, and there's these little tweaks that make us just a little bit different from one another, and it creates this sort of individuality. People with the same disorder can present with a range of symptoms, and this is because we have differences in our genetic background. And so what I can do is take a mutant fly and cross it in or mate it with different flies of varying genetic backgrounds. And I can actually see how these different genetic backgrounds change my mutant fly. And we can sort of get a sense of how genetic variation alters disease presentation.
So we can take images of the eyes, and for example, this one is a control eye, and you'll notice that it's really large, and it has this sort of like organization. Versus when we have a mutant, so this is a genetic mutation, the eye is much smaller compared to the control, and it's a little bit more disorganized. And so through this technique, we can sort of measure the area of the eye and see the differences between control and genetic mutations.
My lab has had a long interest in studying rare diseases, and a lot of it has been driven by being approached by families that are living with rare diseases. And so we wanted to try to use our expertise to help bring some therapies and relief to those families.
So our testing process really has 3 phases. One is we ask about the genetic model of the rare disease in the fruit fly. So we make the fly version of the rare disease, and we look for measurable symptoms in the fly that we can quantify and measure. And then in phase 2, once we've identified measurable symptoms, which we call phenotypes, then we expose the flies one by one to a library of FDA-approved drugs and ask which of those drugs make the flies better, which don't have any effect at all, and which maybe make the flies worse. And then in the 3rd phase, for the drugs that make the flies better, we revisit those drugs and test them thoroughly at like multiple doses from independent chemical companies and really cover our bases. And then we report all of that information back to families and foundations and clinicians that we're involved with. For me, working at the bench doing the experiments, I'm blind to the IDs of the medicines, right? So it's drug A, drug B, drug C. So I don't have any preconceived notions of what drug might help this particular disease. So we're really letting the data drive the discovery. If you think of like a butterfly life cycle that people are kind of familiar with, I think, they have the caterpillar stage, right? And then they make the cocoon, and then they close as the butterfly, right? And fruit flies actually have a similar type of insect life cycle where they have a growth phase and then an adult phase. And we do our experiments. One nice thing about our approach is that when the flies are exposed to the medicines, it's not just as adults. So we can expose the disease model to the medicine in the food where the developing larval phase, which is sort of like the caterpillar phase, takes place. So for example, the flies are exposed to the drug in the food their whole life, and then we can measure the symptoms as adults, their whole developmental life. And then we can measure the effect of the medicine in the adult phase.
So we use CO2 to anesthetize the flies, and they can wake up and recover from this. But it allows us to sort different genotypes, which we can see visually, and set up genetic crosses for our experiments. So here I'm just collecting the females for a genetic cross.
You can easily see by eye whether a fly is a male or female, and the males will have this black bottom, and the females will just have a striped back. And so this is really important for setting up crosses, because you're going to need to know what's a male and what's a female for when you set up your genetic pairings. That's a male right here. You can tell by this black bottom right here. And then something like this would be indicative of a female. When they're flipped over, it's a little bit harder to tell. So if you see the backside of the fly, it's really easy, and you could tell whether they'll have that black bottom versus these, just like the striped bottom. And very important for setting up crosses. You don't want all males in a vial and get no progeny.
You can imagine that you get a population of experimental animals at a pretty quick rate. So when we ask about the effect of a medicine on a given symptom or phenotype in the fly, we get a population of flies coming out of that treatment vial that has been exposed to that particular medicine. So when we have a library of 1,520 drugs, we don't just get one fly per drug, we actually get a little population that's been exposed to that medicine. So the statistics are pretty powerful.
Drosophila are incredibly easy to breed. You can take a few female flies and a few male flies, put them in the same vial, and within a couple weeks you'll have hundreds of flies to do experiments on, to just study. And so Because of this rapid generation time, it makes for a really good model to have a large sample set to get really insightful data. The eye will express the same gene that might be causative of a disease for humans. So if there is a change in the eye size, we can infer the same type of biology is happening in the rest of the body. So this is an example of a wild-type healthy Drosophila eye. And as you can see, the eye is bright red, and it has these organized hexagonal structures along the eye, and this is called the ommatidia. And so in a healthy wild-type fly, these are very well organized. These are the photoreceptors of the fly, and they're all fully functioning. All right, and so this is an example of a Drosophila eye that has a mutation in it. And when we make these flies, we essentially knock down a gene of interest in only the fly eye tissue. And if that gene is important, it'll change the size and the quality of the eye. So here you're looking at a female fly eye, and you'll notice that the eye is much smaller in size, and you lose the structure of all of those photoreceptors that are important for vision and their sensing.
We ran a drug screen. So this was when we subjected our mutant fly to a panel of FDA-approved drugs. And what we found was this compound that came out of it. And because it's FDA approved and we work directly with this nonprofit organization, we were able to give the drug directly to patients.
So we tap the flies into a vial, and then we vortex it for about 10 seconds. And then we just place the vial here, and as you can see, these are all walking around, climbing up.
Normally, when you put them down, they'll sort of reestablish themselves and be able to climb back up normal. However, if we have sort of a neurodevelopmental phenotype or a neurodevelopmental disease model, what we'll see is that the flies seize at the bottom of the vial, and they have a really difficult time reestablishing themselves.
You discover something new every day. Even when you think you know what you're going to get, the flies give you something really exciting that you didn't know.
Sorry.
Science isn't all strict rules and guidelines, and in our lab we have a lot of fun, and we'll do a lot of like dancing together and stuff, just to kind of like sometimes you'll be working on one experiment for months, right, and it just doesn't work, or like something keeps going wrong, and then you get like that one key piece of data and like everything comes together.
A lot of times the educational process can feel kind of long, but I can tell you that it's really energizing to feel connected to helping people.
Together, it's just a really great environment to study human disease using a small little fly.
Being a scientist is really rewarding. There's not one particular type of scientist, right? Working in science for a long time, I've met so many different people with different types of approaches, and everyone's welcome.
When you first get results and it's something new, no one else knows about it, and then my brain starts running around in its head trying to figure out what that means, and I get really excited. I think it's really a fun process being the first person to know something. Like, oh, this gene causes small brains when you knock it out. It's really fun. It's not just studying what happens in the fly. We use it as a tool to study us.
It's really important to give patients answers for things. It shouldn't be overlooked how much it means to a patient to know what's causing their disease. And we have the opportunity to do that for patients. It gives them a lot of relief. It gives them a plan. And I think we can really rapidly figure out, not only if the gene is causing their disease, but we can kind of figure out why. And that means a lot to people, to just know what's going on in in their case, and to know that they're seen and looked at, and you know, they aren't just kind of a number out there in the health system, that they're actually people that want to know what's going on with their disorder.
There are so many rare disease patients and rare disease kids that don't have a therapeutic for them. And for most of these, there's no cure, it's just symptom management. And I think one of the most significant findings is that we really are able to go from the lab to patients, and this is done with just a fly. And we have seen from our lab that drugs that we find from our drug screens are actually making a difference in patient lives.
One of the most exciting things is actually being able to go to the patient meetings that they have every year and really meeting the people living with these disorders and kind of seeing the impact of our work on their lives. We talk with them about the biology and also kind of what we're trying to do and how we're trying to help improve their lives, and I think that that's a really meaningful mission for our science, and that's probably the most exciting part of all this, is really kind of meeting the faces behind the rare diseases.
Rare disease research is highly underfunded and highly understudied. And I think it's so important to get more people on the team and more people excited about working on science that you think may not impact people, but really changes the lives of people. Give us more money, please. We want to save the kids. That's it.